guys, today we're going to be doing DNA extraction. Here we are in our indoor growth facility, and our beans are probably going to be bigger than yours, but if you can find 7 to 10 leaves about this size, then you're all good to go. So let's meet back at the lab. Now that we are back in the lab, let's get started. For experimental purposes here in the lab, we use a more technical process, and for more information on that, there is an article on our website that you can read. But today we are going to be using a method that you can try at home with everyday materials. The materials we'll be using today are the 7 to 10 leaves that we collected earlier, as well as a blender, 450 milliliters of water, 2 beakers, a jar, a juice jar that you can find at home, 6 grams of salt, which is about 1 teaspoon, 10 milliliters of rubbing alcohol, 5 milliliters of bacteria soap, granulated cylinder, and you can use a measuring cup at home, a strainer, and something to pick up your DNA with, as well as paper towels. So the first step to this process is making your saltwater solution. So what you do is that you simply add your salt into the water, and you stir until everything dissolves. So here I've measured out 100 milliliters of our saltwater solution, and you just add it into the blender. And next we're going to grab our plant tissue from the freezer. The reason that we freeze our plant leaves is because when the ice crystals form during the freezing process, the cell walls are broken down. This will result in a higher yield of DNA at the end of the experiment. Now that we have our frozen plant tissue here, we are going to add it into the blender. Make sure that everything is submerged into the salt solution. And then you just blend it for 10 seconds. So now our DNA is in solution, and we are going to filter out the debris using a 2-step filter process. The first one, we're going to use a common day kitchen strainer. And now to remove some of the even smaller particles, we are going to filter using a paper towel. So what you want to do is measure out 50 milliliters of your DNA solution. And then pour it into your beaker. The next thing you do is measure out approximately a teaspoon of soap. You just want to add it into the DNA solution and stir slowly. Make sure that you do not create any bubbles. And now we are going to pour the rubbing alcohol into the solution. We have set our rubbing alcohol on ice so that DNA will precipitate better. And you want the alcohol to float on top, so gently tilt your beaker to the side and add the alcohol in slowly. Last step, we are simply going to fish out our DNA. Today we're going to be using a glass hook, but you can find anything at home, such as a bobby pin. So what you want to do is make small circular motions. And now we just put the DNA into the tube. Now we can use this for experiments, but we will have to further purify it. And that concludes our DNA extraction. Thank you for watching.